exam 5 and a final exam, all right? So if you still have questions about this, please let me know, okay? All right. So, just to continue, some proteins are not enzymes, so then the researchers, they revised this to one gene, one enzyme hypothesis to one gene to one protein hypothesis. Why? Because not all proteins are enzymes, okay? Not all proteins are enzymes. You have other proteins who they serve like structural support, transportations, and channels, receptors, antibodies, proteins, they do everything, right? So a set of portions, they are the ones that are enzymes. So that's why they changed this hypothesis. One gene, one enzyme, one gene, one protein hypothesis. So this one gene will code for one protein. And this protein may be an enzyme, or maybe an antibody, or maybe a receptor, or maybe a channel, or maybe like structural support, whatever it might be, okay? So not all proteins are going to be enzymes. And the same thing, not all enzymes are proteins. The majority of the enzymes are proteins, but there is a subset of enzymes that they are actually from RNA.
From what?
RNA.
Oh.
RNA. So then they changed that one, right? Because proteins are composed of either one polypeptide or many polypeptides, and each one will have their own gene. So then they changed that to one gene, one polypeptide hypothesis. But then we refer to the gene product as proteins and as polypeptides. So right, so transcription translation. In prokaryotes, transcription and translation is coupled, so they happen at the same place, okay? Because they don't have a nucleus. So here you have the bacteria, the prokaryote cells, you have their DNA, and then you have transcription into RNA, the messenger RNA. Then from that one you have the translation, making the polypeptide. And that polypeptide will fall into the protein, make an enzyme, or a structural support, whatever it may be, okay? So key point, transcription and translation, they are coupled in prokaryotes because they do not have a nucleus. In eukaryotic cells, transcription and translation, they are not coupled, they are uncoupled because they have a nucleus. So transcription happens inside the nucleus. You have here the DNA, transcription to the pre-messenger RNA, and then into the RNA processing to the mature RNA, mature messenger RNA. And then, as we mentioned in the first couple of modules, it will go from the nucleus into the prophylor to find the ribosomes, or it will into the cytosol and find the free ribosomes, and that it will take the translation, okay, making the polypeptides. Okay. So transcription, translation, uncoupled in eukaryotic cells. Now, whenever you go into the codons, these are triplets of nucleotides. We flow information from gene to proteins based on a triplet code. So that is a series of non-overlapping 3 nucleotide words. So that is a triplet code, codons, okay? So 3 nucleotides non-overlapping, and that will code in this like triplets of nucleotides, the codon, that will code for an amino acid, okay? These 3 nucleotide words. That will code for one amino acid. So these words of a gene are transcribed into complementary non-overlapping 3 nucleotide words of messenger RNA. So you have the messenger RNA, right, a set of codons, 3 nucleotides, and each of these 3 nucleotides it will code for one amino acid. Non-overlapping, that means that you cannot take one nucleotide from the previous codon. Okay, so non-overlapping. So then these are going to be translated into the amino acids, and then that will make the polypeptide. So here's what you have, the example. So you have here the DNA template strand, and then here you have the DNA coding strand. Always remember this, key point, okay? If you are sleeping, please come back. This is a key point, it will be on the exam. Yes. Huh? What was that?
What's gonna be on exam?
This, okay? Coding strand, it is the one that is from 5' to 3'. Always look for that. That is the coding strand, okay? You can get the DNA template strand, and from that get the coding strand. But if you have the coding strand, no need to do that. No need to find the complementary nucleotides. Okay, if you have the coding strand, then that is telling you the code. That is the code, right? So you just transcribe that into RNA, the messenger RNA. So notice this messenger RNA, it is identical to the coding strand except that the T is changed to the U, USL. But it is the exact same thing, just change the T's to U's. Whenever you see this on the exam, that I give you this DNA, just look for the 5', 3', and write that. That is your RNA, identical, and just change the T's to U. That's it. Identical, coding strand, okay? I have seen students making some crazy gymnastics. Once I give them DNA, they give me the overlapping, and then they change the T's, and then they make another change right there, and at the end, I don't know what they're giving me. Right? It's like they're giving me this, and then making this, and then forgot this, and then change the T's. It's like, what are you doing? Okay. So, 5', 3', coding strand. Copy that. It's the same thing. That is the coding strand, so that is the RNA. Just change the T's to U's. Transcription, like that. And then translation, this is the codon, okay? Triplet nucleotides. 3 nucleotides, 1 codon, 1 amino acid, okay? Then the other codon, 3 nucleotides, and that's another amino acid. Non-overlapping, so that means that this is 1 codon, the other codon will not take these 3 because this belongs to this codon. That is a non-overlapping. It is a friendship, okay? It's written in this friendship where these codons are non-overlapping, and that is translation, okay? Notice this, that the 5' coding strand DNA, 5' messenger RNA, that corresponds to the N terminus of the polypeptide. And the 3' corresponds to the C terminus of the polypeptide. Okay? So make sure you know that. So you remember that.
I have a question.
That should be right there. 5' 3', that is the N terminus, and 3' is the C terminus. Okay. Once you know here, and these are the peptide ones. Yes?
Can you go back? I have a question on that. So remember on one of the IRATs, there was a question that you did have to flip it. How is that worded? Like, what is the DNA template strand?
So on that one, I was just asking you, okay, so here I give you this sequence of DNA, right? Give me the anti-parallel.
I think you said opposite.
So you give me the one that is complementary.
Okay.
But from that one, you have to be careful in give me the right direction, 5' to 3',
Okay.
right? So, there. So if I give you this, right? If I give you the top one, so I ask you, give me the direction, or give me the anti-parallel sequence of DNA. So then you give me the complementary one, but give me that in the right direction, 5' to 3'. Okay. On the question, I might ask you, put 5' to 3', give me the anti-parallel, you will give me this, 5' to 3'. Okay. So that one is just, I'm asking you, give me the anti-parallel, the complementary nucleotides for the DNA.
Okay.
Right direction, you take care of that. On this one, I'm asking you to transcribe and translate this DNA sequence. Okay? So we'll take the 5' to 3', coding strand, identical, change T to use, that is RNA transcription, then from that, translation. 1 codon, 1 amino acid. 5' N terminus, 3' C terminus. Those will be on the exam. Please make sure that you know that, right? Make sure that you take notes of that.
Are you gonna give us a chart of the CD protein?
Hmm?
Are you gonna give us a chart like?
Just. So you guys can do that. So then you have the template strand, that's what I mentioned, the template strand. And template strand is always the same strand for a given gene. The translation mRNA based triplets, those are called codons, or in the 5' to 3' direction. That's what I mentioned there. Yes?
Can you go back? How do we know that it's gonna be those specific ones and not?
Yes, so that's what she asked, the table. So I will give you the table, right? So you can use that. Once I give you this, then from this column, you will go over the table and find out which amino acid it is. That will be good. You don't have to remember that. Only know the start codons and the stop codons. Those are the ones you have to remember. Okay, so that's the same thing that I mentioned. Codons along the messenger RNA molecule are read by translation machinery in the 5' to 3' direction, and each codon specifies the amino acid, one of the 20s, to replace corresponding position on the polypeptide. Okay, so this is the table right there. Okay, so I'm going to give you this. So for example, let's say this one, UGG. How would you find that one, right? So here you have this. So you go U, that's right there. G, that's right there, and then G, right there. UGG. You will find that amino acid right there. Okay? So first, second, and then third. Okay? Let's do AAG. So here you do find A, and then here G. Right there. AAG, amino acid, right there. Okay? Make sure you know how to use this table to find the codon and the amino acid. I will give you the codon, you find the amino acid. The start codon, AUG, that always know. Okay? In case that I ask you, here's this DNA sequence. Start DM, the process of translation. Sorry, I will give you. Here's the RNA sequence, the messenger RNA. Okay? Here you have the messenger RNA. Start the process of translation. Okay? I might ask you translate this RNA sequence, so you translate everything. But if I ask you translate this messenger RNA where it is start codon, so that means that in this RNA sequence that I give you, you have to find first the AUG, and that is the start met, and then from that you will continue on reading the RNA, the messenger RNA, and adding the polypeptide, adding the amino acid to make the polypeptide. Okay? Does that make sense? Yes. So either I ask you to translate the whole sequence that I give you of RNA, or I ask you to translate from this whole RNA sequence would start once you see the start codon. Two different ways. Okay? All right, so all the 64 codons that were deciphered in the mid-1960s, and of the 64 triplets, 61 codes for the amino acids. The 3 triplets are the stop signals to the translation, and this generic code is redundant. That means that more than one codon may specify for a particular amino acid. So here you have this, right? AUU codes for this amino acid, but at the same time this codon, AUC, codes for this amino acid, and at the same time this AUA codes for this same amino acid. So it is redundant, okay? You have 3 codons that code for the same amino acid. It is redundant, but it is not ambiguous. It is not ambiguous in the sense that no codon will specify for more than one amino acid. So that means that this codon will not code for this amino acid and this amino acid. Okay, it's not ambiguous. It is redundant, but not ambiguous. So from this table, make sure you know, yes, the start codon, AUG, and also know the stop codons, UAA, UAG, and UGA. Those stop codons, make sure you know that, right? So UAA, you are awesome. UAG, you are great. And UGA, you get an A. Okay? Make sure you know those 3 stop codons.
It's not gonna be a chart. It's not gonna be a chart on the chart.
Yeah, there will be on a chart, but if I ask you questions, in a particular question, multiple choice, when you see this, you will have to know that. Okay. What will happen for translation if this sequence, it has one of those? Then you will tell me, then we'll stop translation. Okay. All right, so that's what I mentioned right now.
Can you go back to the chart? So what you're saying is on the exam, it's not gonna say like stop next to the ones that are stopping. It's not gonna say, okay, gotcha.
There will be that, but you have to know, right? For
Okay.
example, if I give you a multiple choice question, this I will give you if I ask you to do translation. Okay, this would be better. But if I ask you the multiple choice question, where I don't give you this, but I ask you what will happen to a protein, let's say that it has been mutated, and now the new codon it is UAA. It was UAC, but not because of mutation, it is now UAA. What would happen? Right? So you will have to know that it's a stop codon, so that would, what would happen? The process will halt, it stop, okay? That will be a shorter protein. That will be a non-functional protein, okay? Because it stopped before we're supposed to stop because of this mutation. So it will be a shorter protein and it will be a non-functional protein, okay? So just know these stop codons and know what will happen to them in case that you will see them in the sequences.
61 code for amino acids.
Right, so evidence of revolution, you have here the genetic codex now universal because it is conserved in prokaryotes and eukaryotes, including humans. So that means that one organism can express genes from another organism. So for example here, instead of a plant, expressing a firefly gene, or this pig expressing a jellyfish gene, so they're able to fluoresce, right? So it's universal code. They use these expression of proteins using the same way, right? Transcription, translation.
Wow.
All right, so transcription, let's just look a little more detailed. This is not going to be like really detailed. You will see that like in upper level classes in molecular biology and so forth. This is just like surface level, right? So transcription basically is this. Initiation, you have here the DNA, double stranded. You have here the promoter, data box, and then the RNA polymerase will bind to the promoter region. Bind, and then that is the start point, and then it will transcribe this transcription unit, okay? So it will do that by binding and then unwinding the DNA, remember how helicases did it, once you will open it up, and then it will start making the RNA transcript, okay? So we'll use the template strand to make the exact replica of the coding strand. Here is making it right here. It's going to replace this. But it will not have the T's, it will have the U's, the uracil, okay? So we'll keep making that. So notice this red one, it is 5 prime, it's the exact replica of this one, the coding strand, except that it has uracil instead of timing. So as you keep doing that, the elongation process is going downstream, and then it will reach the termination point, okay? Once it reaches the signal, then it will fall off, and then you have the completed RNA transfer. Okay. Notice this enzyme RNA polymerase can start from de novo, right? From scratch. The DNA polymerase for DNA elongation, it needed the primer, it needed primase to put the primer. So now the DNA polymerase can hatch, can lock, can bind to the primer and then elongate. This RNA polymerase does not need such a thing. It can start from scratch, from the novo. Just bind to the promoter region, and then starts to unwind, and then to elongate, and then it terminates. Okay?
So the white is the stable?
This one?
Yeah.
This one is the RNA polymerase. What's stable? From the following?
Yeah.
From the previous?
Yeah.
No. The
Oh.
stable was a precursor.
Oh,
The minimum media. The enzyme was going to grab the minimum media and use and convert that into the own ingredients, right? Those are like proteins. This one is transcription. Okay? Any question on transcription? No. So key points to remember. Remember the key players. RNA polymerase. What does it do? Binds to the promoter region, elongates, makes the exact replica of the coding strand, except that it has U's instead of T's. And then it will complete the transcription. It will follow. Okay? Starts in novo, start from scratch. Doesn't need to have a primer. Okay, RNA modification. So this is like the pre-mature messenger RNA. It needs to be mature messenger RNA. How it does that? So here you have some RNA modifications. One, it is splicing. So we remove the introns and connect together the exons to put it together. So it can code for the actual amino acid that it needs to. That DNA that is in between those exons, those are the introns. So it needs to be removed. Okay? Those are like hotspots. Those are good for like switching different exons, so it can give you variety of different proteins. Then you have the poly A tail and the 5' metal cap. So these are the 5' and poly A tail is 3'. So that is for protection of the messenger RNA. Then translation. So once you have this mature messenger RNA, then the ribosome will bind, and then it will start making the polypeptide. Okay. For that one, you will need the transfer RNAs, the tRNAs. This basically mediate this amino acid addiction to the polypeptides. They are the ones that bring it. So notice here, this is just RNA holding in this loop, hold together by hydrogen bonds. And notice here that this tRNA has this anticodon, okay? So it will bind to the codon. The anticodon will bind to the codon, okay? So it's a match, okay? So you have here the messenger RNA, it will bind to the anticodon, and this tRNA here is basically will have the amino acid. So these are like two ways to be specific that you are bringing the right amino acid. One, match anticodon with codon. One. Two, tRNA with amino acid. This tRNA with this anticodon will bring this particular amino acid.
Is there only one anticodon? Only one?
For every tRNA, yes. So everything tRNA will have its own anticodon that it will bind to the particular codon from the RNA. Okay? And it will have this particular amino acid. Okay? So two ways to be specific. So here you have the ribosomes. I think I mentioned this before, that there are some differences, bacterial and eukaryotic ribosomes. So because of that differences, you can actually use some antibiotics, right? They use antibiotics and they will target the bacterial ribosomes without affecting the ribosomes. These antibiotics, whenever you get some infections, you get these antibiotics, they're going to target this process of transcription in bacterial cells, but not yours. Okay? Really, really key. Really, really key. So here you have the picture of the ribosomes. You have the small subunit and then the large subunits. Here you have these different sides, the EPA. The A is the A side, the amino acid tRNA binding side. Here you have the P side, peptide tRNA binding side, and then here you have the E side, the exit side. Okay. So it binds here. And notice here this tRNA, anticodon, matched codon, has amino acids. And here, if you want polypeptide, it will change here and will keep going. It will add in the amino acid. And then here the E side will follow without the amino acid. So notice that two ways of being specific. For this tRNA, it has this anticodon, it will match the codon, and this tRNA will bring this particular specific amino acid. Okay. All right, so once you have this, so first the AUG, right? The start codon, as I mentioned that before, make sure you know that one. So once you have the RNA, you have here the AUG, anticodon, right there, match, boom, stop. So here's the assembly of the ribosomes. It begins the small ribosomes unit, and then you will need GTP energy. GTP is basically the cousin of ATP, and it will use that as source of energy to assemble the whole ribosome complex. Okay, so now the large subunit comes, and it will start translation. From that one, you will bring the next tRNA, match it, and bring that a particular amino acid. Again, it would require GTP to bind this tRNA into this anticodon into the codon. Okay, this tRNA with the messenger RNA. Once it binds, then it's going to move the polypeptide and then move as well here the growing polypeptide, and then require energy again to release that tRNA that is no longer has the amino acid. Okay, so two steps where you need the energy, the GTP. One, here in the binding, recognition, and two, in the release of the tRNA. Okay. Once you're done, then here you have the stop codon, UAG, you are great, UAA, you are awesome, UGA, you get an A. Once you have that, then stop. Stop codon, here you have the release factor, and then you will here release the free polypeptide and release the tRNA, and then it will require energy as well to release, dissociate the whole complex. Translation. Yes?
The start and stop codon will be on the mRNA, not the tRNA.
That's right. That's right. And the messenger RNA. The tRNA will be anticodons. Yes. All right, so now we go to the last section, and that will be mutations. So here you have these different mutations. These are changes in the genetic material of the cellular virus. You have like 4 mutations, so like chemical changes in just one or few nucleotide pairs of a gene. And then you have this change of a single nucleotide in a DNA template strand that can lead to the production of polymer protein. Just by one change of a nucleotide nucleotide, you can change the whole function or the structure of the protein. For example, here you have the wild type hemoglobin. Here you have the sequence, DNA sequence. Here you have the coding strand, GAG. Here you have the messenger RNA. Notice it's identical. Just grab the coding strand right there. There is no T's right here, right? So there is no need to change the T's to U's because there is no T's right here. Some people, because they see T over here, they see the need to change all T's to U, and then they will use that and change this T to U and give me the wrong thing. Okay, so do not do that. Coding strand right here, convert that into the RNA strand, messenger RNA. It is identical. If there is no T's, then it is identical. Don't change anything. Then from here, it will give me the amino acid translation. Okay. Whenever you have here single point mutation, nucleotide, this is changed. So now here, instead of an A, I have a T. And here, instead of a T, I have an A. So they are just changed. Because of this, now the RNA changes. Now there is a T over here in the coding strand. So now here it's going to be a G, U, G. That will give me the wrong amino acid. And that will have an effect on the structure, and that will have an effect on the function of this protein. I will see the case study for next session. So right now, just to see these different types of mutations. So you can have, like, for example, nucleotide per substitution, or one or more nucleotide per insertions or deletions. For nucleotide per substitution, so this basically replaces one nucleotide and its partner with another pair of nucleotides. So just change one, right? And that's it, just one change. Okay. So these different types of mutations, one, it is the silent mutations. Silent mutations, as the name implies, silence, there is no effect, right? It is as if there is no mutation right there. So this one has no effect on the amino acid that is produced by codon because the redundancy genetic code. Remember that it is redundant or not ambiguous. It is redundant, so it has no effects. This is known as the wobble effect. Okay. Why? Because notice here. This CUU codes for this amino acid. CUC codes for this amino acid. Notice that, right? It's redundant. So if I change the U to a C, I will still have the same amino acid. That is known as the wobble effect. These two are conserved, are the same. If I change the third one, wobble effect, mutation on the third one, no effect, silent mutation. I will still get the same amino acid. But if I change the fourth or the second one, then I will have different amino acid. Makes sense. So silent mutation, wobble effect, redundancy of the codon. So I will get the same amino acid. Okay? That's the example right there. Okay? Notice here you have CCA, CCG, the G changed to an A, but because these two are the same, the last one is changed, but I still get the same amino acid. Wobble effect, redundancy, okay? Silent mutation. Missense mutation, that is the one that it changed the DNA, but now it will give me an incorrect amino acid. Okay? Missense, the wrong amino acid. And substitution mutations are usually missense mutations. So notice this, what happens here, this one, it changes the first nucleotide in the codon. Remember, if you change the first one or the second one, then yes, it will bring the wrong amino acid. Here, it's supposed to give me glycine, but now it's giving me serine. Missense, the wrong amino acid. If it gives the wrong amino acid, it will change the structure of the protein, and if it changes the structure, it will change the function of the protein. Okay. The other one is the nonsense mutation. So this change of the amino acid, it will now code for the stop codon. Okay, that is a nonsense mutation. And if it now codes for the stop codon, so that will lead to a shorter protein and to a non-functional protein. As you can see here, right? This mutation right here, from this T to an A, and now here we have here the UAG. Now that is stop codon. We're supposed to be growing, but now it stops, it halts. So now you have a shorter protein and a non-functional protein.
When it stops, you don't transfer any of the
That's right, because it stops, right? It halts. Yes.
So if it stopped, how come there's still amino acids?
Codons, right?
Sorry, codons. Yeah, why
There's
is
still
this?
codons, but now here's the stop signal.
Oh, okay.
The stop signal truncates everything, stops everything. Remember, whenever you have the stop codon right there, if this is changed, right? Let's say that it's doing the whole process, right? But then here, it has a mutation. It's supposed to continue elongating the protein, but now it's a mutation, and here the RNA, it goes for the stop codon. It's supposed to be over here, but now it is here. If it is mutated and now it has a stop codon, then it will do this. This stop codon will bring the release factor and release everything. So now this free polypeptide is actually shorter than it's supposed to be. And because it is shorter, then it's no longer be functional. It will not be able to do what it's supposed to do. Okay?
I guess I was confused as to why the strand of codons kept continuing after UAG.
Yeah, because this is the RNA.
Right.
So transcription takes place, right? Transcription is not
But
dependent on
it's the
stop codon.
translation that's gonna stop right
The
there.
translation stops.
Okay.
Yeah. Transcription makes this until it reaches termination factor and it will be released, right? But it will transcribe whatever is here. In this case, if this is the mutation, the stop codon, it will continue to transcribe RNA polymerase.
Okay.
But the ribosomes, once it reaches this, it will stop. It will not continue the translation, the elongation of the polypeptide. Okay. Alright, so that is whenever you have the changes of DNA. But also you can have like insertions or deletions. These are really dangerous because these are going to cause a frame shift in the reading of these codons into the polypeptides. So for example here, if you have this normal right here, wild type, right? But then you have here nucleotide pair insertion. So you have here now an extra A. Notice what it adds, right? The extra A right here, it adds a codon. So now this insertion causes an immediate nonsense. So a stop codon, okay? Terminates right away. Insertion, it can have either the immediate nonsense or, let's say here, in this case, it's a deletion. It misses this, it deletes this A right there. So now because it is deleting this A, so it's now going to grab the next nucleotide from the next codon. So now the codon changes, right? It's going to grab the next nucleotide from the next codon, and then the next codon will grab the next nucleotide from the next codon, and then it will change the whole amino acid with that. So it will be an extensive missense. So this will be wrong, therefore this will be wrong, this will be wrong, this will be wrong, because now they're grabbing the next nucleotide from the next codon. So that's why insertion or deletion are really detrimental. It can cause an immediate nonsense, or it can cause an extensive missense. Okay? So everything is wrong. It might be shorter, it might be longer in the wild type, but definitely it's non-functional. And then here, this is the whole nucleotide pair deletion. If it is the whole codon missing, so there is no frame shift, but it's just missing amino acid. Okay? So this is missing, so now we'll just continue to do translation, the next codon, and adding the next nucleotide, or the next amino acid. Okay? So this one is missing amino acid. So here's the table, make sure you know all of this. Type of mutation. The type of mutation, make sure you know that if it is a silent mutation, no effect, right? What will affect redundancy of the codes? If it is a missense mutation, bring in the wrong amino acid. If it is a nonsense mutation, bring in the stop codon, so shorter and non-functional, and the nucleotide pair insertion or deletion. If it is added or deleted, it can cause an immediate nonsense, or it can cause an extensive missense, or it can cause whenever you are missing just one or having an extra one amino acid. Okay? So nucleotide pair substitution and nucleotide pair insertion or deletion. Make sure you know that. Okay, just to close, here these are like mutagens, so you can have like spontaneous mutations can occur during DNA replication, recombination or repair, that is normal. You have like mismatch repair, but sometimes you have those combination of those mutations, spontaneous mutations. But you also have mutagens, which are like physical or chemical agents that they can cause these mutations. So these researchers have developed methods to test mutagenic activity to chemicals. And most cancer-causing chemicals, those are carcinogens, are mutagenic, and the converse is also true. So that means that these carcinogens are going to cause mutations, and these mutagenic, those are cause mutations, agents, are going to be carcinogens. So, what is a gene? We have